The patented QuanDx Yin-Yang probes are double-stranded DNA with different lengths. The longer strand is labeled with a fluorophore, and the shorter strand is labeled with a quencher. When the two strands are close together, the probes are non-fluorescent. In the presence of the target DNA strands displace the negative probe strands along with their quenchers, and the fluorophores become fluorescent. During real-time PCR, this process occurs during the annealing step with target DNA PCR products. Signals are collected at this annealing step. Yin-Yang probes show high specificity for multiplex PCR reactions. We are going to talk about leukemia fusion gene detection in the next few slides. Leukemia is cancer of the blood cells, usually affecting white blood cells, which causes them to not behave normally. There are four main types of leukemia. Leukemia can occur in either the lymphoid or myeloid white blood cells. Cancer that develops in the lymphoid cells is called lymphocytic leukemia. Cancer that develops in the myeloid cells is called myelogenous leukemia. The disease can be either acute, it begins abruptly and is usually short-lived, or chronic, it persists for a long period of time. Acute leukemia involves new or immature cells called blasts, which remain very immature and cannot perform their functions. The blasts increase in number rapidly and the disease progresses quickly. In chronic leukemia, there are some blasts present, but they are more mature and can perform some of their functions. These cells grow more slowly, so the disease progresses gradually. Based on these findings, leukemia is then classified into one of the four main types of leukemias: acute myelogenous leukemia, or AML, chronic myelogenous leukemia, CML, acute lymphocytic leukemia, ALL, or chronic lymphocytic leukemia, CLL. In addition to these, there are other types and subtypes of leukemia. Leukemia is related to fusion genes. Here's an example of two fusion genes. Prognosis prediction, treatment options, and minimal residual disease monitoring are reasons to detect leukemia fusion genes. Fusion genes can be formed from chromosomal translocations, inversions, and deletions. There are more than 100 MLL gene-related rearrangements, and 90% of the MLL fusion genes are the 10 types listed here. Karyotype analysis is a laboratory technique where the chromosomes from one cell are visualized under a microscope to investigate the total number and structure of the chromosomes. Cells are obtained from an individual and are viewed during metaphase, a stage in cell division when our chromosomes are condensed. The chromosomes are stained with a dye, resulting in a banding pattern of light and dark stripes, known as G-banding. The patterns are specific, allowing us to identify each chromosome. This technique has both pros and cons. Fluorescence in situ hybridization, or FISH, is a cytogenetic technique that uses fluorescent probes to bind to only those parts of the chromosome with a high degree of sequence complementarity. It is used to detect and localize the presence or absence of specific DNA sequences on chromosomes. This technique also has both pros and cons. Multiplex real-time PCR to detect specific fusion types has become a popular technique. There are also difficulties in designing multiplex real-time PCR tests. The Leukemia Fusion Gene Q30 Screening Kit is an RT-qPCR-based assay for the detection of leukemia-associated fusion gene transcripts found in total RNA from whole blood or bone marrow samples. Ready-to-use cDNA synthesis reagents and qPCR primer probe mixes are included in the kit. The test detects more than 140 breakpoints for the 30 fusion genes in 8 tubes simultaneously. Sample fusion gene and control gene transcripts are co-amplified in each reaction and identified by specific fluorescent probes. Step 1. Select 30 common fusion genes and design the 8-tube reaction system. Step 2. Design primers and probes for the specific fusion genes. Use one probe and multiple primers for those with multiple formats. The schematic at the top shows an example of the GUSB internal control design. The schematic on the bottom shows the detection of BCR-ABL1. Primers are depicted by horizontal blue arrows. Breakpoints are shown by vertical arrows. Step 3. For assay development, construct positive plasmids for different fusion types. Step 4. Assay optimization to detect all selected fusion genes. Serial dilutions of plasmids have been performed to determine the sensitivity as being within 10 to 100 copies per reaction. These slides show examples of positive patient sample results. In summary, the Q30 kit highlights are shown here. The QuanDx Q30 screening kit can be used as a powerful tool for leukemia patients in diagnostics, prognosis, and treatment monitoring. One example of the clinical validation data is shown here. A total of 195 clinical samples were screened with the Q30 kit, and 98 samples were determined to be fusion gene positive. The positive fusion gene samples were checked with DNA sequencing, and the results were a 100% match with Q30. The Q30 fusion gene screening kit detects more than 140 breakpoints, while the AML kit screens 72 and the ALL kit screens 71 breakpoints. Rapid results can be obtained within 3 hours. The QFusion system provides a sensitive, specific, reliable, and cost-effective tool that makes it easy to perform routine screenings of leukemia fusion transcripts. The AML Fusion Gene Screening Kit detects 16 common AML fusion genes in one 8-tube reaction. The ALL Fusion Gene Screening Kit detects 16 common ALL fusion genes in one 8-tube reaction. Please visit www.quandx.com for more information.